A research group at MIT tracked cognitive flexibility in adults across a 20-year window. The study, published in 2019 in Nature Neuroscience, followed over 1,200 participants from age 25 through age 45. They measured one specific thing: the brain's capacity to rewire itself in response to new information. The technical term is synaptic plasticity. The finding was precise. Peak neuroplastic capacity occurred between ages 25 and 32. After 32, measurable decline. By 38, the average participant showed a 47% reduction in the speed at which new neural pathways formed compared to their baseline at 25. Not memory, not intelligence. The physical speed at which the brain rewires itself. That is the starting point. Now, everything in this video follows from that number. Most discussions about brain aging focus on memory decline or cognitive disease, Alzheimer's, dementia, pathology. But the research documents something occurring decades before disease appears, a progressive narrowing of the window during which the brain can efficiently reorganize itself. The popular narrative suggests you can keep your brain young through puzzles or learning new languages. That narrative is not wrong. It is incomplete, because what the MIT study and dozens of others like it reveal is that neuroplasticity is not just about what you do. It is about the biological substrate that enables plasticity in the first place. And that substrate changes on a schedule the research can now measure. The question becomes, what is the mechanism? Why does the brain's capacity to rewire itself decline in a predictable pattern across the third and fourth decades of life? And is that decline purely chronological, or is it driven by something measurable at the cellular level? The answer exists in published literature. Multiple independent research groups have converged on the same biological system. A 2021 study from Stanford University School of Medicine examined synaptic density in healthy adults across age ranges. They used high-resolution brain imaging to count the number of active synaptic connections in the prefrontal cortex. The prefrontal cortex is where executive function lives: decision-making, planning, learning new patterns. The study found that synaptic density peaked at age 27 and declined by an average of 1.4% per year thereafter. On sample size, 914 participants. The decline was linear, predictable, and it correlated directly with performance on neuroplasticity tasks. The more synapses lost per year, the slower the participant's brain adapted to novel cognitive challenges. That gives us the what? Synaptic density declines. Now we need the why. Research published in 2020 in the journal Cell Metabolism identified the upstream driver. A team at the University of California, San Francisco measured mitochondrial function in neurons across different age groups. Mitochondria are the energy factories of the cell. Neurons are among the most energy-demanding cells in the human body. A single neuron can contain up to 2 million mitochondria. And the UCSF study found that mitochondrial efficiency in neurons declined by approximately 32% between ages 30 and 40 in healthy adults. The measurement was ATP production per mitochondrion. ATP is the energy currency every cell uses. When mitochondrial ATP output drops, the neuron has less energy available for the most energy-intensive process it performs, building and maintaining synaptic connections. Neuroplasticity is not a psychological concept. It is an energy problem. A 2022 meta-analysis published in Frontiers in Aging Neuroscience reviewed 41 studies on synaptic plasticity and mitochondrial health. The pattern held across all studies. Declining mitochondrial function preceded declining synaptic density by an average of 3 to 5 years. The mitochondria lose efficiency first, Then the synapses that depend on that energy begin to prune. The brain does not lose plasticity because you stop learning. The brain loses plasticity because the cellular machinery that supports synapse formation and maintenance begins to operate below the energy threshold required to sustain it. That is the mechanism. Everything downstream follows. The biology works like this. When a neuron forms a new synaptic connection, it is performing construction at the molecular level. The process is called synaptogenesis. The neuron has to synthesize new proteins. It has to transport those proteins to the exact location where the synapse will form. It has to assemble receptor molecules on the cell membrane. It has to maintain ion gradients across that membrane so the synapse can fire correctly. Every single one of those steps requires ATP. A lot of it. Research from the Max Planck Institute for Brain Research calculated that the formation of a single new synapse requires approximately 10 billion ATP molecules. When mitochondrial output declines by 30%, the neuron does not stop forming synapses. It forms them more slowly, and it prioritizes which ones to keep. The brain begins to triage its own plasticity. A study published in 2023 in Nature Aging examined this triage process directly. Researchers at Yale University School of Medicine used fluorescent markers to track which synapses were pruned and which were maintained as mitochondrial function declined in aging neurons. The finding? The brain preferentially prunes synapses associated with novel or infrequent patterns and maintains synapses associated with well-established routines. This makes metabolic sense. Maintaining a synapse you use every day costs less energy than maintaining a synapse you formed recently but have not yet reinforced. The result is that as mitochondrial efficiency drops, the brain becomes better at doing what it already knows how to do, and worse at integrating genuinely new information. This is not a failure of effort. This is resource allocation under energetic constraint. The implications extend beyond learning speed. A 2021 study from Johns Hopkins University tracked emotional regulation in adults across the same age window. Emotional regulation is a neuroplastic process. When you experience an emotional trigger and choose a different response, you are building a new synaptic pathway that competes with the old one. The study found that participants over 35 showed significantly slower progress in cognitive behavioral therapy compared to participants under 30. Same therapy, same adherence, different biological substrate. The researchers controlled for motivation, life stress, and therapeutic alliance. The variable that predicted outcome was baseline mitochondrial function in prefrontal cortex neurons. The neuron's capacity to rewire depends on the energy available to rewire it. One more layer. Research published in 2022 in the journal Science examined what happens to synaptic plasticity when mitochondrial function is artificially supported. The study used a compound called nicotinamide riboside, a precursor to NAD+, which is a molecule required for mitochondrial ATP production. NAD+ levels decline with age. Supplementing with NAD+ precursors has been shown in multiple studies to restore mitochondrial function toward more youthful levels. The Science study gave healthy adults aged 35 to 50 either nicotinamide riboside or placebo for 12 weeks. Then they measured synaptic plasticity using the same tasks from the MIT study. The group that received the NAD+ precursor showed a 23% improvement in neuroplasticity task performance compared to baseline. The placebo group showed no change. The mechanism was clear: restore mitochondrial ATP production, and the brain's capacity to form new synapses improves. This was not a cognitive enhancer. This was addressing the energetic bottleneck directly. Research has identified a natural compound that supports this neural and mitochondrial mechanism at the cellular level. It is not a drug. It is not a treatment. It is a natural formula built from published research on brain and cellular energy metabolism. Yet, if you want to understand how it works, the link is in the description. This is a sponsored recommendation and individual results vary. The framework this creates is different from the standard conversation about brain health. The standard conversation asks, are you experiencing cognitive decline? The research reviewed here asks a different question. Is your brain operating at the energy level required to maintain the plasticity you had at 27? Most people never think about this until they notice a problem. But the mitochondrial decline that underlies synaptic pruning begins years before subjective symptoms appear. A 2020 longitudinal study from the University of Pennsylvania followed adults from age 30 to age 50 and measured both mitochondrial function and subjective cognitive complaints. Mitochondrial decline became measurable at age 32. Subjective complaints did not appear until age 41. 9 years of invisible decline. The labs measure disease. This mechanism operates below that threshold. If this gave you a new way to think about your biology, leave a comment below. I read every one. And if you found this useful, the single best thing you can do is follow the channel. We decode a new mechanism every day. Everything you need is in the description and in the first comment. The research on neuroplasticity windows connects directly to measurable biological systems you can assess. Before you go, we built a free 2-minute biological wellness assessment that reviews 4 body systems and gives you a personal pattern score. The link is in the bio, in the first comment, and in the description. 2 minutes. Manchip puts numbers to everything we just discussed. Recent research from 2024 has begun mapping the genetic factors that determine individual variation in this neuroplastic decline. A study published in Nature Genetics identified 3 gene variants associated with the rate at which mitochondrial function declines in neurons. Participants with all 3 protective variants showed half the rate of synaptic density loss compared to those with none. Sample size, over 4,000 participants across 5 countries. This finding suggests that the neuroplastic window is not purely chronological. It is genetically modulated. 2 people at age 35 may have significantly different synaptic plasticity profiles depending on which variants they carry. The study also found that lifestyle factors, most particularly aerobic exercise and caloric restriction, could partially override genetic predisposition. Exercise increased mitochondrial biogenesis, the creation of new mitochondria inside neurons. Caloric restriction activated pathways that improved mitochondrial efficiency. Both interventions shifted the neuroplastic curve. A separate 2024 study from the Salk Institute examined synaptic plasticity in adults who maintained high cardiovascular fitness versus sedentary controls. The fit group showed synaptic density levels comparable to individuals 7 years younger. The mechanism traced back to a protein called PGC1-alpha, which regulates mitochondrial biogenesis and is upregulated by sustained aerobic activity. When PGC1-alpha levels rise, neurons produce more mitochondria, and those mitochondria operate more efficiently. The result is more ATP available for synaptogenesis. The neuroplastic window does not close on a fixed schedule. It closes as a function of the energy environment inside the neuron. Another research direction involves the role of inflammation in neuroplastic decline. A 2023 study published in Brain Behavior and Immunity measured inflammatory markers in cerebrospinal fluid and correlated them with synaptic plasticity performance. Participants with elevated levels of interleukin-6 and TNF-alpha both pro-inflammatory cytokines, showed significantly reduced performance on neuroplasticity tasks regardless of age. Chronic low-grade inflammation, the kind associated with poor diet, poor sleep, and chronic stress, appears to accelerate synaptic pruning. The proposed mechanism is that inflammatory signaling interferes with the brain-derived neurotrophic factor pathway. BDNF is a protein that promotes synapse formation and maintenance. Inflammation suppresses BDNF. Less BDNF means slower synaptogenesis and faster synaptic pruning. The study controlled for age, education, and baseline cognitive ability. Inflammation was an independent predictor of neuroplastic capacity. A fourth line of recent research has focused on sleep architecture and synaptic homeostasis. A 2024 study from the University of Wisconsin-Madison used calcium imaging to observe synaptic pruning in real time during sleep. The brain prunes synapses during deep sleep as part of a process called synaptic homeostasis. This is normal. The brain strengthens important connections and removes weak ones. But the study found that when deep sleep is chronically disrupted, the pruning process becomes dysregulated. The brain prunes more aggressively and less selectively. Synapses that should have been maintained are removed. Over time, this leads to a net loss of synaptic density beyond what would occur with normal aging. The researchers measured this across 6 months in adults aged 35 to 50. Those with fragmented sleep showed a synaptic density decline rate twice as fast as those with consolidated sleep. The mechanism linked back to mitochondrial stress. Fragmented sleep increases oxidative stress in neurons, which impairs mitochondrial function, which reduces the energy available for maintaining synapses. One of the more interesting findings comes from research on bilingualism and neuroplasticity. A 2023 study published in the Journal of Neuroscience compared synaptic density in lifelong bilinguals versus monolinguals across the same age range. The bilingual group showed a slower rate of synaptic decline. The hypothesis is that managing 2 languages requires constant activation of executive control networks, which sustains higher baseline synaptic density in the prefrontal cortex. This is not about learning a new language at 40 to reverse decline. And this is about sustained cognitive demand across decades, creating a different synaptic architecture. The bilingual advantage was most pronounced in participants over 45. At younger ages, the difference was minimal. This suggests that sustained cognitive complexity does not prevent synaptic decline, but may slow its trajectory. Finally, research published in early 2024 in Cell Reports examined the role of the gut microbiome in neuroplastic capacity. The study found that specific bacterial strains in the gut produce metabolites that cross the blood-brain barrier and influence mitochondrial function in neurons. Participants with higher levels of butyrate-producing bacteria showed better performance on neuroplasticity tasks and higher synaptic density on imaging. Butyrate is a short-chain fatty acid produced by gut bacteria when they ferment dietary fiber. It has been shown in animal models to improve mitochondrial efficiency and reduce neuroinflammation. The human study replicated these findings. Higher dietary fiber intake correlated with better neuroplastic performance in adults over 35. The effect size was modest but measurable. This opens a new intervention point, modulating the gut microbiome to support brain mitochondrial health. And all of these findings converge on a single framework. Neuroplasticity is not a fixed trait. It is an emergent property of the neuron's energy state. That energy state is determined by mitochondrial function, inflammatory environment, sleep quality, physical activity, and even gut microbiome composition. The neuroplastic window does not close arbitrarily. It closes when the biological systems that support synaptogenesis fall below the threshold required to sustain it. The research suggests that threshold is modifiable.